So first off, could you just explain what NCI-MATCH is?
Sure. NCI-MATCH is a version of a platform trial, and it's a platform that basically on which sit dozens of so-called basket trials or umbrella protocols. The basket trials is, I think, the most commonly used term, and basically the way that term was apt for how we constructed NCI-MATCH is that each of the arms of the trial was a pairing of a molecular feature, commonly a genetic feature, but some molecular feature of cancer assigned to a specific therapy, but with no limitation in terms of cancer type. So open broadly, each of the therapeutic arms open broadly to all cancer types, but where the constraint was on a common molecular feature. And that idea of a basket trial, so pairing molecular feature with a therapy, that's relatively new enterprise still in oncology. So it's been investigated. Concepts like that have been investigated going back now, maybe the earliest example about 2013, but have become more and more common over recent years. Recall that we designed NCI-MATCH in 2014 going into 2015. So at the time, it was still a relatively young concept, and our idea was to try to broaden it to simultaneously investigate all of the available kind of pairs of molecular feature and drug. So that is what NCI-MATCH is.
Great. So now could you explain the key highlights of the recent study results regarding tumor gene makeup?
So this recently published report in Journal of Clinical Oncology is the first analysis of the genetic landscape, if you will, in the population of patients that we screened for NCI-MATCH. So for the first roughly 2 years of the study's conduct, as opposed to the more recent 2 getting on towards 3 years of sort of follow-up clinical investigation, in that first 2 years, we were performing central molecular testing. So we set up a network of 4 laboratories, all of which were using the same next generation sequencing platform. And all of them were sort of qualified and essentially evaluated for their ability to produce similar results using the same platform across 4 laboratories. So all of the samples, just under 6,000, we used the number 6,000 in describing this cohort because that was the a priori target population. And the number in the end fell a little short of 6,000, not because fewer than 6,000 were considered for the study, but rather the number that were evaluated found to be eligible at the first pass to go into the centralized molecular testing was this just under 6,000, but 6,000 was the target. In any case, we aimed to centrally screen 6,000 advanced cancer patients, had to have had a cancer type for which no available effective therapy existed, which for certain rare cancers is unfortunately still the case to this day, or if they had a cancer type for which there are available therapies that are truly effective, they must have received those previously and had them unfortunately not be successful prior to being considered for NCI-MATCH. So that kind of level set the degree to which the study was targeting a population who we would say were refractory to all of the available therapies. So, this represents really the first analysis of this kind, of a genomic analysis of this large number of patients who were clinical trial candidates and who had either failed all the available therapies or for which no effective available therapy existed, broadly across all cancers, as opposed to just focusing on one cancer type. And so that type of analysis has not previously been done. There have been other published reports, certainly of individual tumor types. Of course, the 2010-2015 era was notably populated with a number of analyses like that, including the Cancer Genome Atlas. But that, distinct from this analysis, was really focused on patients' primary cancers. So their initial breast cancer, their initial lung cancer, their initial colon cancer, not the metastatic form of the disease, and certainly not those patients who had actually seen multiple types of therapy. Those were treatment naive, if you will, or untreated patients in that analysis. So this is at the other end of the spectrum. This is really what types of patients, what types of cancers, and really critically, what molecular features basically come through as being refractory to the available treatments, right? Because you can imagine that along the way, there are patients who had surgery and were cured by surgery plus adjuvant systemic therapy of some kind, drug therapy that cured them. We would never have seen those patients, right? So there's a kind of almost a selection out, leaving behind then essentially the genomic landscape of what remains as the unmet need broadly across cancer. And so I would say that really was the key highlight of this report, is what that genomic landscape is of the remaining unmet need as we look at cancer now from a molecular perspective.
So why are these findings so important, or what are the implications of the findings?
We'd say that for this analysis, which is separate from continued reporting of the individual treatment arms and how successful or unsuccessful any of those treatments might have been across the group of patients who were treated with a given molecular feature paired with a therapy, the importance of this genomic landscape map, if you will, is that it really allows planning to be done now going forward. So we, of course, used this map to guide how we conducted NCI-MATCH, but now it really informs not only the next generation platform trials that the NCI itself is launching, been designing, and now on the verge of launching, but really any other type of any other approach comparable to this that aims to try to take any of these molecular subtypes and now do follow-on investigations with either individual new drugs, new regimens, or kind of mixtures of multiple drugs. When we designed the study, we had some insight as to what the frequency might be with which we would find some of these alterations, but we were surprised both with more frequent observation than we had predicted, and then commonly less frequent than predicted, and that's critical. I mean, otherwise it really is very difficult for one to prioritize, and then even just to understand the feasibility of moving forward in some of these populations to try to deliver therapies that are tailored to these molecular types or molecular signatures.
So what would you say is most important for medical oncologists to understand regarding these recent findings?
I think the word that I would highlight that we used repeatedly in this manuscript is actionability. And by that I mean actionability of next generation sequencing. So this type of molecular analysis has become widespread, certainly over the life cycle of NCI-MATCH, and in fact dating back maybe just a couple years before then. Rather than doing individual molecular tests in certain cancer types for which there either are approved drugs or clinical trial investigational drugs, the idea of just doing a single test that would capture all of those, both standard of care markers and investigational directed markers, if you will. That approach certainly has its efficiency, but in terms of being able to just find kind of all of this information all at once. But really, I think what hadn't been described previously is really in this population of trial eligible, kind of real world patients, if you will, as opposed to patients who have primary tumors subjected to surgery who may or may not ever have recurrence of their disease, that in this population, which is the very group in which we're doing most of our clinical trials with novel therapies, now we have an aggregate view of the likelihood of finding molecular alterations that point to currently FDA-approved therapies, and that was about 18% of the NCI-MATCH population. We didn't offer them these known effective treatments because, of course, they're available. So that those patients, if they hadn't already received them, would then, it would be suggested that they go on to receive those. But then, in addition to that, we had another 17% in the published analysis here of the population for which we had investigational treatment arms. And so, just simply doing the addition of those two numbers, that defines a rate of what we're calling actionability, which will change over time and has already been changing since we began conducting this study, of roughly 35% of patients who have something for which we can say is either known to be effective, FDA approved, or promising based on human evidence combined with preclinical evidence. And so I think the field has really been lacking simply a number to say, okay, if we do this type of testing, what is the utility of it? What can we do with the information? What's the likelihood I'm going to be able to point my patient to some follow-up therapy that's guided by this type of analysis? So I think that's really the critical benchmark that comes from this. And my hope and expectation, and I think even in 2020, I could say we're witnessing that number is rising. How quickly it's rising and how steadily it will rise over time, I think that's difficult to predict, but it is going up. So I think we set sort of a 2015 to 2017 sort of benchmark, and now as new discoveries are made that offer new treatments for patients with unique molecular features, that will just add to that number.
So for patients who may not understand these findings, how would you describe the key highlights in lay terms?
I think that this study underscores the degree to which doing molecular testing, particularly for patients who have advanced cancer, so metastatic cancer, I think can comfortably be said now to be the standard for cancer patients, regardless of their cancer type. That this type of analysis, I think, can be declared to be a component of standard care, both to navigate to available treatments that are already FDA approved and those that are in clinical trials. In other words, simply knowing what type of cancer one has by its site of origin, lung cancer versus breast cancer versus colon cancer, I would say we can say now with this report, this type of analysis, that's not enough to really guide treatments for a great many of our advanced cancer patients. And I think that's perhaps the most important point, is that in addition to knowing what type of cancer one has, you need to know a bit more about its molecular makeup so that patients and doctors can make the very best treatment decisions.
So what are the next steps for the study moving forward?
So the trial has actually continued on after the phase reported in this manuscript, where we were doing quote unquote centralized screening. After that 6,000 number had been reached, which was the a priori target population, we still had a significant number of our treatment arms that were still looking for patients, particularly some of the ones that were looking for hard to find or low frequency molecular alterations. And so we kept the study open. And what we did was we then expanded beyond the 4 laboratories that we had been using for our centralized testing. We kept those laboratories operational, but we expanded beyond that to any commercial or academic laboratory that could show themselves to be proficient in performing next generation sequencing. So we had this qualification process that we established. That's actually been independently published, that process. And then we relied on their testing to identify patients for those remaining treatment arms that were still looking for patients. That process is still ongoing as we speak. So there's been this slower rate of uncovering those patients with quote unquote standard genetic testing now to complete the therapeutic aims of the study. So we had the centralized screening phase, and now this kind of more gradual process of finding these, fairly rare patients. And so that's, that's been the up till now and still ongoing version. I would say next steps for this study are further analyses of the molecular features combined with the treatment outcomes to try to understand where to go next, for each of these treatment and molecular pairings. So in other words, which ones should actually just continue to accumulate more patients and more treatment outcomes over time, because there's something promising already suggested in the data, versus those cases where we have to move already to combination approaches to try to improve outcomes for some of these same patient subpopulations. There is a study referred to as combo match, which is one of the National Cancer Institute supported studies that is being mounted with that type of mindset. So I think for this specific study, it's a deeper dive. What we presented so far with this 6,000 patient analysis is a high level view. There's granular data that readers can access, not just from the paper itself, but from the now publicly available data to do some of that diving themselves. But we will do more analyses to try to map out what we think are the paths forward for each of these subpopulations.
So that's all I had for you. Is there anything that I didn't ask that you think is worth mentioning in this space?
I think the two other points that I would raise about NCI-MATCH that are clear successes that I think point towards a new way of doing clinical investigation are that we probably forever going forward need to be thinking about this mode of investigation based on molecular features and not purely based on the kind of legacy notion of cancers defined by their site of origin. So I think that the concept of then doing this sort of the basket trial, but really a collection of them, if you will, it has great efficiency for doctors and patients, and that's the most important point. This was a trial that was adopted by 1,100 sites, many of them academic medical centers, and many of them private practices and practices that are part of networks, so smaller centers, where adopting this trial was a very efficient way of having essentially multiple trials available all at once. So one study that contained multiple or many. And that efficiency, I think, is a clear success. And we need to keep options like that open to try to keep clinical trial availability at the highest possible level broadly across the entire cancer patient community. The other final point that I would emphasize is that we were able to attract a large number of patients who have rare cancer types, for which studies are generally not mounted. So there are many rare cancer types. So when you add them together, depending on how you want to define rare, you can easily come up with a number like 20% of all advanced cancers are rare cancers, but just many different types that, when you put them together, they're 1/5 or 20% of all cancers. And yet we don't really attend to that group of cancer patients with dedicated studies for their individual rare cancer types. So it's for rare cancers that it's particularly important to be thinking in this molecular definition of cancer diagnosis, as opposed to just the cancer as we look at it under the microscope or describe its origin, if you will. And that's been a real unmet need. And this trial all at once was quite a success in terms of being able to now investigate broadly across rare cancers. And I think that's a moving forward, next step element that I and many others involved in this study that think might be one of the really important enduring legacies.